Of course, exogenous compounds, be they man-made or found in nature, aren't the only way to modulate excitation and inhibition. Diseases, such as myasthenia gravis or congenital myasthenic syndrome, can also influence the delicate excitation-inhibition balance that is necessary to ensure proper function of our nervous system. Myasthenia gravis is an autoimmune disorder that leads to serious muscle weakness and fatigue. The weak muscles are caused by circulating antibodies in the blood that block or even lead to the destruction of acetylcholine receptors at the postsynaptic neuromuscular junction. As a result, acetylcholine can't bind to the receptor and excite the postsynaptic cell, and it can't stimulate muscle contraction. Myasthenia gravis is classified as an autoimmune channelopathy, since it features antibodies directed against ion channel proteins that are naturally present in the body. The causes of the disorder are still unclear, but various medical treatments exist in the form of acetylcholinesterase inhibitors and immunosuppressants, which allow most patients to have a normal life expectancy. Congenital myasthenic syndrome, or CMS, is an inherited neuromuscular disorder caused by defects of the proteins that make up the neuromuscular junction. The effects of this disease are similar to those of myasthenia gravis, but the big difference is that in congenital myasthenic syndrome, it's not an autoimmune disorder. Postsynaptic defects are the most frequent cause of CMS and often result in abnormalities in the nicotinic acetylcholine receptor. More than half of the currently known genetic mutations associated with CMS are found in one of the 4 genes encoding the acetylcholine receptor subunits. If impaired, these faulty receptors can't participate in the proper excitation of the muscle fibers during neurotransmission, which results in muscle weakness and fatigue. Although the symptoms are similar to myasthenia gravis, immunosuppressant treatments used in myasthenia gravis are generally not useful for CMS patients. Alternative forms of drug treatments for CMS do exist, however, including the use of acetylcholinesterase inhibitors, such as quinidine or fluoxetine, depending on the specific subtype of CMS that's predominant in the patient.